The topic of the day is eukaryotic cell division. What you're seeing here is referred to as the cell cycle, which is, as it suggests in its name, a cycle, meaning there is no beginning and there is no ending, so we're going to have to pick a place to start. If you follow the red dot here, we're going to start right here. And that cycle is going to continue around, and eventually what we're going to talk about now is going to end right here. In other words, we're avoiding the section referred to as cell division, and we're going to deal with everything up to that point. That whole period of time from where we're going to start to where we're going to end today is collectively referred to as interphase. Interphase is the portion of time in the life of a cell when that cell is doing whatever job that cell is supposed to do. All of our cells are specialized. So brain cells need to act like brain cells and lung cells like lung cells, etc. During all of interphase, those cells are performing those jobs. You will notice that the cell cycle, and specifically interphase, is divided up into smaller portions. We have the G1 phase, the S phase, and the G2. We're going to work our way through those three portions of interphase and talk about the special functions that take place during each. We're going to start off with the G1 portion of time. Now, the G of G1 simply stands for gap. It's a gap of time. And you'll notice in the grand scheme of things, it is within the cycle occurring right after a cell has just divided. Therefore, it's very small. It just got split in half. So you'll notice in terms of things going on, one of those things that is going on during the G1 portion of interphase is that the cell needs to grow. It needs to get back to a normal-sized cell. Also, number two, the production of cell organelles. Again, you have to imagine that the cell has just finished dividing. One cell became two. That means that all parts of the cell were divvied up amongst two cells. All the contents of the cytosol, all the ribosomes, all the mitochondria, all of those organelles were split in two. And so this new cell, as it goes through G1, not only is it going to grow, it's going to reproduce all those quantities of organelles that it needs in order to do its job. Of the three portions of interphase, probably the most significant would be the S portion. The S simply stands for synthesis. Synthesis means to make something. And so in this case, as is stated here, what's going to be made are chromosomes. We're going to replicate all of our chromosomes. So in a human cell, there happens to be 46. Those 46 exist right here, but as the S portion of interphase progresses, all 46 of those human chromosomes are going to make an exact copy of themselves. Why? Because this cell knows that eventually, if we continue through this cycle, we are going to get to this period of time called cell division, in which one cell splits into two. Each cell that results needs a complete copy of all of the chromosomes, and therefore, back here during the S phase, that spare set is going to be made. Thinking along those lines, we need to look at this picture right here. You'll notice two terms, monad and dyad. Of course, the prefixes are key here. Mono means one, di means two. At the beginning of the S portion of interphase, we have 46 monad chromosomes. What does that mean? Well, let's look at the picture over here to the left, and let's just look at the left side of this picture. Where I have the red dot going right now would be one monad chromosome. The whole point of the S phase is to make an exact copy. So what you're seeing on the right side here is the replica of what's on the left. Now, we tend to think of this, of course, as two separate chromosomes because, one was the original and one was the copy. And yet, we always want to say that humans have 46 chromosomes. So instead, we have gone from having 46 monad chromosomes to eventually having 46 dyad chromosomes. Those dyad chromosomes are held together here in the center through something called a centromere, and the two halves of those dyad chromosomes are referred to as chromatids. They are exact copies of each other. They're so exact that they are specifically referred to as sister chromatids. Finally, we get to the G2 portion of interphase, and there's not a whole lot to say about this. The cell is preparing to divide. Okay, so we're getting ready for that cell division process. Before we can actually do that, though, let's look at what a cell would look like while in interphase. It doesn't matter whether it's G1, S, or G2. If we look at a plant cell, which is what you're seeing here at the top, it's going to have this basic look about it. You can see a cell wall over here, another one over here. This is the nucleus of the cell, and you can see a dark mass within the nucleus that is the nucleolus. Down here on the bottom, we have an animal cell, and it's very similar. Got a cell membrane, no cell wall. You've got a nucleus. And other than that, there really isn't much to see. We have this representation here that should show you basically the same stuff. You got the cell, you've got an intact nucleus, you've got some genetic material inside that it has represented here. And one thing we didn't see in the previous pictures, you can see centrioles that will later be involved in cell division. Before we can actually get into the how cells divide, we need to talk about the why cells divide. The first three of the list here are pretty basic and straightforward. Think about yourself when you were younger, maybe 5 years old. Now think about yourself today. You're clearly bigger today than you were when you were 5. That growth has been allowed because cell divisions occurred. You're made out of more cells today than you were when you were 5. Point number 2, the repair of damaged tissue. You fall down, you skin your knee, maybe somehow you break a bone. During any of those situations, when repair is necessary, we need to make new cells in order for those repairs to occur. And so again, cell division would be necessary. Third, we've talked about the fact that we have cells that are programmed to die at a certain point. They only have a certain lifespan. We refer to that as apoptosis. And so as we have cells die, and the easy example here would be skin cells, every time we shed a skin cell, we need to replace it with a new one, and so cell division is necessary. The last two are a little bit more complicated, but both involve the growing size of a cell. I mentioned earlier that cells grow. DNA overload refers to the genetic material inside the cell. I mentioned earlier, we have in human cells 46 chromosomes. Those 46 chromosomes control everything about the cell's activity, everything about the cell doing its job. They are responsible. And when the cell is small, that's good, it works. But as a cell continues to grow and get bigger and bigger and bigger, those 46 chromosomes struggle in order to maintain control of the cell. And so the easy solution is divide. If the cell gets too big and then divides, it becomes small again, and those 46 chromosomes can again control the activity of the cell. Think along those same lines for number 5. The problem here is the surface area to volume ratio. The surface area to volume ratio deals with, again, cell growth. As the cell grows, the surface area and the volume obviously both grow with it. The problem is that the volume actually grows faster. So let's say that the volume of the cell has grown 10 times its original size, but at the same time, the surface area only grew 6 times its original size. If a cell is 10 times greater in volume, well, then it's going to need 10 times the nutrients. It's going to produce 10 times the waste. Well, how do those nutrients get into the cell and how does the waste get out? through the cell membrane, and that's represented by the surface area. The problem is the surface area is only 6 times bigger than it used to be. And so the volume being 10 times bigger, requiring 10 times the nutrients and producing 10 times the waste, the surface area just can't keep up. There's not enough space for nutrients to enter or for waste to be exiting. So again, the solution is the same. Have the cell divide, the cell gets smaller, problem solved. Finally, in terms of eventually talking about cell division, we have 2 main parts to that process. There is, as it is listed here, mitosis and cytokinesis. Together, they compose cell division. The focus is simply different between the two. When we think of mitosis, it's all about the chromosomes. Now remember, they're the most important thing about a cell. And so when a cell is going to divide, it becomes very important for the chromosomes to all get split up properly. So the total focus of mitosis is going to be on those chromosomes, while cytokinesis is going to be about dividing up everything else, all the contents of the cytosol, all the organelles, the mitochondria, the ribosomes, etc. They all have to be divvied up between 2 cells. That leads us into talking about cell division.